So it's a real pleasure to welcome Dr. Richard Bethlehem as our speaker today. So obviously Richard is known to many of us already, but for those who haven't met Richard, he's a research associate at the Cambridge Brain Mapping Unit and also director of neuroimaging at the Autism Research Centre, obviously both here in the Cambridge Psychiatry Department. Richard's research focuses on understanding both typical and atypical neurodevelopment, really by combining both neuroimaging data and also transcriptomic and genetic data. So I believe you're going to talk to us today about your recent work on brain charts for the human lifespan. And without further ado, I will hand over to you, Richard.
Thanks, Sarah. That's a very nice introduction. Yeah, so like Sarah said, my background is a little bit in autism research and in development, but the work I'm actually going to be presenting today, I think hope stretches a little bit beyond that. So I'll be presenting like some of our recent work, but I'm also going to take a little bit of a trip down memory lane, at least for me, for some work we did sort of leading up to this, to kind of put it a little bit into context. And so just to sort of preface it with saying, like, a lot of the examples I'll probably be giving are likely going to be related to autism. Because that's where I first started out with this work. But I like to think these methods are much more widely applicable. So these are just examples of a methodology we're sort of trying to develop. So yeah, I'm going to be talking about brain charts for the human lifespan, which is a project I wouldn't say completed recently, but which we recently published and which is very much sort of ongoing work. So if there's time today, I can also talk a little bit about what we're sort of planning to do next with it. But before we get into all of that, it might be good to just start with what kind of led up to this project. And so I think that specifically takes us back to essentially two questions. Why brain charts? So why do I think brains are an interesting thing to look at? And a lot of this comes from like I said, my background in sort of looking at atypical and typical neurodevelopment, and basically from this idea, which was, I think, very elegantly put forward by John Gilmore a couple of years ago, and there's another paper from Thomas Paus in Nature Reviews Neuroscience as well, kind of outlining that, like, in early development, there are these massively rapid changes happening in the brain. Like it grows absolutely incredibly fast in the first couple of years of life. And a lot of this sort of intersects with where we think some of these developmental disorders might start, or where we first start sort of seeing symptoms, or we first start sort of diagnosing these things. And to me, that's always been sort of an obvious point that maybe there's actually some relation between these two things. Like, obviously, ideally, we'd be able to sort of measure these changes quite early on using ideally neuroimaging, and maybe make some potentially predictions of who's sort of on track to developing in a typical or an atypical manner. And I should say, in the right plot here, I've added ASD, developmental delay, and intellectual disorder, because sadly those weren't in the Meyer and the review, but this is roughly, I think, the point at which some of these can be diagnosed and tend to be diagnosed. And I should probably also say that, well, if you're a geneticist, you're probably going to disagree with all of this and just say that all of this is to a large extent predetermined genetically. And there's something to be said for that, but I think it's still relevant and interesting to see how some of those genetic predispositions might actually lead to changes in the brain. So this is just a very long-winded way of saying, I think brains are extremely relevant for our behavior, for psychopathology, and I think there's a lot of value in trying to sort of map those changes and trying to map essentially when an individual might end up on a trajectory of sort of atypical development. Now, the second part of that question is why charts? So obviously, like, charts is something we see a lot in clinical medicine, but it's not something we generally use a lot in sort of neuroimaging research. And to me, this kind of goes back to sort of this canonical example of the pediatric growth chart as basically an instrument that provides some kind of standardized assessment and does it in a way where we can say something meaningful about individuals. And as you'll see later in my talk, I think especially that second part is extremely relevant when we start looking at whether it's developmental or neurodegenerative conditions, because in practice in a clinic, you're not treating groups, you're treating individuals. And I think that's sort of an essential step in developing some of this methodology, is that we actually end up with instruments where we can potentially say something meaningful about individuals, but not groups. So this is a bit of a divergence, I think, from the more classical research, where we're just comparing the average of one group with the average of another group, which is often great to delineate sort of common, like, neurobiology, or to figure out what might be sort of the common thread that sort of groups these people together. But in practice, if you want to say something about whether an individual is developing typically or atypically, we need something that is much more tailored to that specific individual. And so this is where a lot of my initial research started. Obviously, I'm not the first to sort of even think about this concept that typical and atypical maturation are important to look at, and that we probably need some kind of reference to be able to say something about atypicality. Like, in order to basically define what is atypicality, we obviously first need to know what is typical. Now there's a really wide range, I think, of literature that has kind of tried to map some of this out. And I think a lot of, at least the work that's influenced me quite a lot, is work from Armin Rasnahan, sort of mapping out volumetric changes, looking at sex differences in those volumetric changes, and also the work from Nico Dozenbach, looking at sort of functional connectivity changes and trying to figure out what does the sort of typical trajectory of development of some of these variables that we can derive from neuroimaging, what do they really look like? And then the Sowell et al. Paper is probably one of the earliest examples of where they use neuroimaging to try and really assess sort of developmental trajectories in this. So this is all just to say, I'm definitely not claiming originality in this idea that we should be looking at developmental changes, and that there's value in us first establishing what the sort of normative reference, if you will, looks like. Another thing that I think is sort of helpful to contextualize that idea of having a normative reference, I think is quite nicely illustrated in a paper from quite a few years ago now from Adriana Di Martino, where she basically outlines these sort of various scenarios of how we can conceptualize atypical maturation. And this is done in her case in the context of autism and in the context of looking at functional connectivity, whatever measure you use for it. But I think the sort of infographic that she provides actually gives a nice overview of possible scenarios, whether you're looking at autism or another disorder, or whether you're looking at functional connectivity of another disorder. So what she shows very nicely is that you can think of the sort of typical trajectory as depicted by this blue line, and then there's various scenarios where how atypicality might present itself, as it were. And I think what, at least for me, one of the key messages from this is that also depending on kind of where you are potentially on this atypical curve, if you only have a single point of measurement, it's actually kind of hard to delineate. So if you can think of, if you can see my mouse, if you can think of you, for example, measuring a person sort of like in early development, and it looks like they're falling really far below the sort of typical curvature, it's very hard to say, well, is that because they have some halted development? Is it because they have some delayed development, but they might catch up later? Are they failing to mature altogether? Like, that single time point isn't really giving you that much information. And so, again, a very long-winded way of saying there's a lot of value in having a longitudinal assessment. But the big caveat to that, at least in neuroimaging, is that that longitudinal assessment needs to be valid over time. You want to make sure that whatever you're measuring over time is standardized in some way and is sort of standardized against a specific reference point. So you can think of in a scenario, for example, where someone gets scanned one day in Cambridge, a couple of years later, they might have moved cities and they get scanned in London, but there's not really like a way of telling whether their shift in relation to the sort of typical reference curve is purely due to the fact that they've just been scanned on another scanner in another city, or whether there's actually sort of a different trajectory happening. And so in that respect, I think there's also a lot of value in having a sort of standardized instrument to sort of measure these things. And with MRI, that is a little bit more complicated than how the traditional growth charts are done with just the measuring tape, because there's a lot of sort of technical complexities and covariates that we obviously have to consider in order to be able to do this. So now, in an ideal scenario, and this is sort of the research scenario that kind of started some of these fields, we'd be able to sort of use these some of atypicalities or these deviations from the norm to actually dissociate individuals, to say something, maybe even at some stage use them for a diagnosis. And so what I'm plotting here is sort of on the left, what you're seeing is a lower dimensional representation of a whole bunch of variables that we can measure in the cortex. In this case, it's looking at cortical thickness, but we've made similar plots looking at other properties that are measured with MRI. And then using a sort of very simple data-driven type clustering approach to see, can we actually dissociate two different clusters? And in a very ideal scenario, I mean, here you can see we can associate two clusters in the left-hand plot. And in an ideal scenario, our fancy data-driven algorithms would be able to pick up on like who's an individual with autism, who's an individual that doesn't have autism, or in any other sort of clinical context. In practice, however, you see something that looks much more like the picture you see on the right, because that is actually the sort of real diagnostic labels that we have in this data set. And you can see that it's extremely heterogeneous. And so the problem arises from this is that actually like this sort of conventional approach of trying to compare one group with another group might not always be capturing this heterogeneity. You might be able to see, like you can see in these sort of histograms that are or in these density plots that are plotted around it, there might be some slight shifts. If you do a conventional sort of means comparison, you might see that on one of these axes, maybe the group is a bit lower than the other group, and you find some subtle differences, but it doesn't really reflect the sort of heterogeneity, and it doesn't really pick up on anything that is unique to any of these individuals in this distribution, probably. And so that's where this first idea comes from, of trying to find instruments that tell you something that's unique about the individual. And we've sort of collectively, I think we call this normative modeling, which is basically the idea of positioning an individual relative to a normative reference point. And so this is work we did a couple of years ago using specifically the Abide data set. So the Abide is a big publicly and openly available autism data set of about, I think it's about 800 individuals with a diagnosis and 800 individuals without, if you combine both of the Abide release 1 and 2. So it's a decently powered data set, at least for what we're used to working with normally in neuroimaging. And basically what we did with this data set is we tried to establish sort of what does the normative level of variation look like in a given brain region for a given variable. So looking at cortical thickness in frontal cortex, for example, you can think of that as an example. Taking all the sort of neurotypical individuals from this data set and just kind of plotting like how did they look? What's the variation like across the different age range? And then what we did next is we computed this, what we call the W score, which is very analogous to a Z score, with the only difference is that you're computing it relative to an external reference and not relative to the reference point in the data set itself, which is effectively just a measure of, for a given individual, for a given brain region, for a given variable, what's the sort of amount of standard deviations that this individual might be away from the normative reference curve. So this is a way of standardizing this level of variation across different subjects, and it gives you an individualized metric. Obviously, like when we're doing this approach, we were dealing with lots of different brain regions, lots of different phenotypes, which makes it a little bit sort of complicated to actually say something that is specific about the individual as a whole. And so what we first did is we tried to sort of summarize that pattern of atypicality, because we had one of these scores for each region. And basically just computed a very simple ratio, saying like the ratio of regions that we thought were atypical, so more than 2 standard deviations away from the sort of normative level of variation, versus the number of regions that were sort of within that range. And so when you compute that, like if that ratio is above 0.5, it means to have relatively more regions that are atypical than regions that are sort of in the typical range. And so that is quite a broad pattern of atypicality, you would say. Like the majority of the brain regions would be considered sort of in the extremes of the distribution. And when we did this in the Abide data set, I personally was very surprised to find that I think out of 800 or so subjects, we only find about 12, 13, depends a little bit on sort of which measure you're looking at, individuals that are actually sort of above that threshold, that have actually more atypical brain regions than they have typical brain regions. And so to me, that's quite striking. And it's also, in a way, sort of quite informative to the way that people are finding sort of broad group differences. But to some extent, maybe these group differences are in practice kind of driven by only a small set of individuals. And so what we did next is we basically took a step back and basically took this conventional approach of how people normally do this type of research and ran a very straightforward case control linear mixed effects model. So this is the model I was talking about before. We're just comparing the means of 2 groups. And in this case, again, it's looking at cortical thickness. And we find that there are some regions that are thicker or thinner in individuals with autism relative to neurotypical individuals. However, if we, out of this massive group of about 800 subjects, if we take out just those 12 or 13 subjects that have this highly atypical pattern, we see that the results actually change quite drastically. And so to me, this is quite alarming. And it means effectively that potentially a lot of the things we know about broad sort of underlying neurobiology that is generic in autism might actually be biased by a small subgroup that shows sort of highly atypical patterns that are maybe not very representative of actual individuals in that group. So this is, again, a long way to illustrate that maybe moving away from that conventional case control analysis will lead to something that is a little bit more clinically significant, but also actually accounting for the fact that there are these sort of extreme patterns would probably give you something that is a little bit closer to what is really the sort of the common threat in some of these individuals. Now, sort of towards the end of this project, we also realized there's a lot of issues with the approach we took, with what we know from previous research and what we know from previous sort of benchmarking. And so I think the obvious thing is just that the age range of a lot of these data sets is very limited. So if you think about the sort of first figure I showed, we can see that there's these rapid changes very early in development, and then it kind of, wouldn't say it flattens out, but the variation becomes much more subtle compared to this sort of rapid spurt in early development. And most of the data sets that we have access to that are public, specifically the ABIDE data set, they tend to start from about 6 years and onwards. And a lot of the public data isn't really there in that younger age range. So we're actually missing a very important period in development where we might be able to see either early indications that someone might end up on an atypical trajectory, but also where there might be a lot more variation in the population in general to sort of detect some of these things. And so I think that's a very big caveat to any work. And you see that kind of across sort of previous literature that has looked at establishing some of these normative references, where the age range tends to be from about 6 to maybe 30 here in the Dozenbach paper, or 20 in Armin Resnahan's paper. And even in the ones that have a somewhat wider age range, I think it's a little bit misleading that this one says 0, because again, it starts around actually 6, where the first data point is, going up to 80, but you can see the data becomes a lot more sparse as sort of sample gets older. And it's a very similar pattern in ABIDE, where say after 30, there just isn't that much data. And so that effectively means that you have to wonder how reliable that normative reference really is, because if you're only basing it on very sparse data that isn't really covering the entire lifespan, that probably isn't really covering sort of these rapid changes that we're seeing, maybe that's one of the big caveats. Another big caveat that we obviously, I think everyone's quite aware of nowadays, but it took a while, I think, for that realization really to parse through, is that there's a lot of variation depending on where someone is just measured. So on the top here, I'm showing a paper that at least influenced me quite a bit, from Har et al., I think from 2016 it was, basically showing sort of broad case control differences between autism and neurotypical individuals across the different sites in the ABIDE data set. And overall, they find a significant difference. But actually, if you parse down in all the individual sites, there's actually only 1 or 2 sites where they find significant differences between these two groups. So there's a lot of, let's call it sort of technical variation, or maybe technical noise across these different sites that makes it a little bit tricky to sort of aggregate data together. And then the final bit to say to that is, obviously, all of this data tends to also be estimated from cross-sectional and not longitudinal data. So there's still that question of how stable are these trajectories? How much individual level variation really is there in what these trajectories may look like? Now, I'm not really a statistician. I'm also not really a clinician. Like my solution, and that's why my solution to everything is always to just try and get more data, because that always solves all your problems. And so that's kind of where this brain chart project really started. I would say it started about 6 years ago. The idea started about 6 years ago, but it really got a push about 2.5 years ago with just this little group of people coming together and figuring out ways to mine data. So initially we started with a lot of publicly available data sets, such as the UK Biobank, ABCD, Human Connectome project, and basically anything we could get our hands on. And then we still realized we were missing a lot of data in these sort of crucial periods during development, but also in aging, actually. And we took that initial publicly available data and basically started reaching out to people in our network, people that have published on data, to see if they'd be willing to share data with us, just for the effort of trying to make these reference charts as representative and as robust as they possibly could. Now, as you might have seen from the paper, that's definitely an effort that got a little bit out of hand, to the point where I think Jacob and I probably now live pretty much on Zoom or on our emails, because we got a really overwhelming positive response from lots of people all over the globe sending us data. Also to the point where I've kind of given up on updating this slide, because even though the paper is now out, this is something that still keeps changing. So you can see some numbers in the panel below. I think those are fully out of date. We're probably now sitting on close to 150,000 scans. From, I think it's about 135,000 unique individuals. So there's a little bit of longitudinal data in there. Well, a little bit, about 25,000 scans or so. That depends on what you call little. We have a reasonable, I would say, representation globally of what is available in terms of research data, but you can also very clearly see from the map that it's predominantly sort of Europe, North America. There's something we're actively trying to address by seeking out data sets that are being collected in either more remote locations or just in parts of the world that we don't tend to hear much about. I think we also have a reasonable distribution or a reasonable amount of data sort of spread out across the lifespan. I was quite surprised to see that actually the biggest dip seems to be between the sort of 30 and 40, 45-year-olds. This is where we don't have that much data. Maybe that's not considered the most interesting age group to look at. But the caveat to that plot is obviously that the sort of peak on the right and the peak on the left are skewed quite a bit by the fact that that contains UK Biobank and ABC, which are just really big cohorts. And maybe also to say that if you think of it from a modeling perspective, then actually if you assume that there are these rapid changes in very early development, then that is probably also the part of the lifespan where you want to have the most data, because that's where you want to be able to pick up on the most variation. And so from this massively aggregated data set, we started really quite simply. So we tried to learn from our previous mistake of trying to do this across the whole brain in a very sort of spatially detailed manner, and then trying to have to deal with how do we put this all back together again at the end. But instead, we started with some very simple phenotypes and really tried to get a modeling framework that would be robust first before trying to extend it to something that was a bit more fine-grained. So we basically started with the major tissue compartments in the brain: gray matter volume, white matter volume, subcortical gray matter volume, and total ventricular volume. And what you see in the top plot here is just raw data plotted on a graph with colored by sex in this case. By biological sex, and you can actually see there's already some curves in there. It doesn't really look like it needs much correction for you to be able to draw like a nice line and to see like what those curves look like. But it did also signal a couple of things to us that kind of influenced the sort of choice of modeling that we wanted to apply to this. So first of all, what you can see in these plots is there's likely going to be some non-linearity in this. It's not just a simple matter of drawing a straight line through this. It's not even a simple matter of drawing a straight line up to a certain point and then drawing a straight line down, which is, I think, what some people might think it would have looked like. You can also see in the top plot that the distribution or the amount of variation itself varies across the lifespan. So there's a lot of variation in certain parts of the lifespan that isn't there in other parts of the lifespan, which means it's something we want to somehow be able to A, account for, and B, actually measure. And then the other aspect of it is that that's something you can't really see very well in this plot, but we also wanted to have a modeling framework that doesn't really make that many assumptions about the data. So that doesn't make an assumption that at any point in the lifespan, this data is going to be normally distributed or not, simply because we also wanted a framework that was flexible enough to be adapted to any future sort of phenotypes. And that landed us on this framework called CAMLSS, which are generalized additive models for location, scale, and shape, which incidentally is also the framework that the WHO recommends you use for growth chart modeling. I'm sure that's not a coincidence. I'm sure they had very similar considerations to what we had, but it was very nice to sort of find that like someone had actually already evaluated this. Although, to put that in perspective, I think at the very end of this very long WHO white paper on statistics for growth charts, they basically say you should be using this, but actually for height and weight, you can also just use linear models because it just does it just as well as like this much more fancy tool. Nonetheless, we stuck with the fancy tool because it also provided us a way to measure some other properties. So what I'm showing in the bottom panel here is essentially these growth curves that we get out of that model. So it's showing not the confidence intervals, but it's showing the different sort of centile lines. So the thick lines are the 50th centile, very similar to what you'd see on a pediatric growth chart. And then the dotted lines are, I think from memory, it's the 97.5 and the 2.5 centile lines. And so you can see that these look quite stable, but you can see that these centile lines are also accounting for the fact that there's different levels of variability across the lifespan. So it's a little bit similar, I guess, to a pediatric growth, but a little bit less in the sense that you don't see really parallel centile lines, because in certain parts of the lifespan, these are going to be much closer than in others. The other thing that this modeling framework allowed us to do is also to actually assess, like, at which points in the lifespan do we see the most variability? And we can see that for some of these properties, like the biggest variability is quite early in the lifespan. For some of these properties, such as maybe the white matter variation, it's a little bit later in the lifespan. And although we didn't really go into this in depth in the paper itself, I think there's a lot of value in sort of following this up to see whether in certain clinical populations, for example, the periods where we have this larger population variation might actually intersect with when people tend to be diagnosed. So this is this idea of essentially like a vulnerability hypothesis that where there is more variation, there's also more room for something to go wrong. So that maybe that's also the period in which we tend to see like psychopathology developing effectively. The other thing that this modeling framework nicely allows us to do is also to look at the rate of growth. So which periods of the lifespan do we see these changes happening the fastest? And again, you, at least for these sort of gross, big brain morphological phenotypes, we see that all of these peaks tend to be very early in the lifespan. It's a bit harder to see, I think, in the ventricular volume one, because it's such a skewed distribution to the right, where it just seems to almost exponentially increase in size, but there are some peaks earlier on. And some of that like skewness is also obviously driven by the fact that all of these are on a log plot. So basically emphasizes the big changes that we see in early development in most of the other phenotypes. Now, as I said, we set out to do this as a very flexible sort of modeling framework. And obviously, once we had that in place, we could also try and throw some other potential phenotypes at it. So we also looked at things like total brain volume effectively, or total cerebrum volume, total surface area, average cortical thickness, and then also looked at some of these regional profiles. And again, I think a very similar picture emerges where we see a lot of these changes happening in early development. But in the case of mean cortical thickness, we even see that the sort of fastest rate of change is actually prenatally, which to me is very interesting to see, but also that the sort of peak cortical thickness is actually around slightly before year 2. And I think that's particularly salient in the context of a lot of people effectively publishing on trajectories in cortical thickness, but not actually measuring it until year 6 and onwards. At that point, you're probably not really measuring a trajectory anymore. It's just almost a linear decline to some extent. And I don't think we would have been able to like identify the peak this closely without really having like that very early data, let alone being able to identify at which point during the lifespan some of these things change the quickest. Then in terms of looking at sort of regional volumes, you see there's quite a bit of variability in terms of at which age these regions have their maximum volume. I think we got a lot of, well, I wouldn't call it flack, but we got a lot of comments on social media when the paper came out saying people that I'm already past the peak of all of this, so clearly it's all downhill for there. This is all obviously volume. I'm not making any claims about what that might mean for your cognitive abilities, but at least when it comes to sort of gray matter volume, we can see that these all tend to peak quite early in the sort of, well, between 4 and 11 age range, more or less. And there is an interesting sort of, I would say, anterior-posterior gradient in that development. And again, we didn't go in depth on this in the paper, but I think there's a lot of interesting follow-up potentially just in sort of delineating that pattern. And I'm sure there is lots of interesting theories about why those different brain ranges might sort of mature at different rates. And so this is just kind of a big summary figure of all of those. It's basically outlining all of the different milestones that we're able to derive from these like whole brain type phenotypes, and then kind of listing like in the circles you see like where the rate of change peaks, so where basically like this property sort of develops quickest, and in the little triangles you see where there's sort of the peak volume or the peak thickness or the peak surface area. And I think what's nice about this is we put this in contrast with this recent paper from Solmira, which I think is in molecular psychiatry, where they basically did a meta-analysis to assess at what age people tend to be diagnosed for all these different clinical conditions. And so that's what you see in these little black boxes. You see this is sort of the age range at which people tend to be diagnosed. What you see in the background in the sort of shaded blue area is actually the age range where we have data on individuals in these groups. And so to me, this is like quite an exciting thing to see, is that actually we have a lot of data that goes way beyond the sort of age range that we normally tend to have, we normally tend to diagnose in. And that I think gives a lot of opportunity to start looking at whether there might be very early deviations that could be picked up with MRI that are not really immediately apparent even in the diagnosis. The other thing which I think is interesting, which is why we put this figure up, is there's obviously a lot of other instruments that measure development, but as it turns out, MRI is one of the few where we actually have data across the entire lifespan, and we look at the same thing across the entire lifespan. So ultrasound is obviously like something that's used a lot in pediatric care, but that kind of stops after birth. Then there's height and weight growth charts from the WHO. They stop at around year 18 or 20. Same with head circumference, that stops even earlier. And there's obviously like reasons for that, in the sense that probably those things don't change that much anymore. But I think at least when we look at MRI, we can map the entire lifespan, but that also means we can actually start comparing sort of deviations from these trajectories at different points in the lifespan. So we could actually, and it's something we've done a little bit, we can actually, for example, compare what does the deviation look like in someone with a neurodegenerative disorder versus someone with schizophrenia? And even though those individuals tend to be diagnosed in completely different parts of the lifespan, because all the values we're getting from these reference points are normalized, they're centile scores, and they're age appropriate, so to speak. So that kind of brings us back a little bit to this idea of getting individualized centile scores and kind of comparing those and seeing whether there's sort of clinical utility in some of them. And I've kind of gone against my own sort of preaching at the beginning of saying we need all these individual measures. We have all these individual measures, but obviously to summarize them, we put them all in one figure. And so here what we're just showing is effectively the distribution and the medians of centile scores across all the different clinical populations that we have access to. Actually, we're only showing them for the 7 cohorts or the 7 clinical diagnostic cohorts where we have more than 500 or so individuals with a diagnosis in our data set. But we actually have, I think it's around 110, 120 different clinical labels in the full data set. But some of those are just much smaller groups, and obviously making this plot for 4 different measures, 2 different sexes and 110 different conditions would have been pretty illegible. But we also, I'll come to this at the end, we also have a web type where people can kind of explore some of these differences in themselves. We chose the 500 as a somewhat arbitrary number because we could actually also do some statistics on that would hopefully be robust. And generally what this graph is kind of summarizing is that we see on average, there's quite a lot of sort of deviation, and we kind of, to a large extent, this figure is also just to kind of confirm things that we already knew. So if you're looking at centile scores, you'd probably expect people with a neurodegenerative disorder such as MCI or Alzheimer's disease to have much reduced gray matter volume because they have neurodegenerative disorder. I think what to me was a little bit more surprising is that in some of these measures, we see almost a similar effect size in schizophrenia. So we see almost a similar relative reduction. It doesn't necessarily need to be an absolute reduction, but we tend to see these individuals sort of scoring in the sort of more bottom half of the centile scoring. Other things that are a little bit more hidden, we get into that in the paper a little bit in the supplements, is I think in some of the developmental conditions, specifically ADHD and autism, we see that the variation, sort of the median differences from a control group are not that big. They are significant, but that's also by virtue of probably having quite a big data set that we can find those differences. To me, what is a lot more interesting, you can see that a little bit in the subcortical volume plot, but you can see it in some of the others, is that it doesn't really look like their distribution is centered around the median of the control population. So the black line is the median of like typical, say, let's call it typical development of the 50th centile line. We can see that there's actually, well, we look at anxiety disorder here, you can see it a little bit in the autism group as well. Where there tend to be there might potentially be some subgroups that score high or low, but if you put all of those people in one group together, that tends to average out a little bit. So I think that potentially is something that is very interesting to follow up on to see like whether there is whether these are meaningful subgroups. And it does to some extent, and this is more speculation than something we've actually tested. It does confirm this idea that in autism we see individuals with micro and macrocephaly, and that might propagate into having high or low centiles in these groups. But if you put all of those 2 groups, if you put both of those groups together with anyone that doesn't have micro or macrocephaly, then maybe that averages out and you don't really find these differences. We also tried to, even though it's only 4 variables, we tried to simplify this a little bit further by effectively trying to create sort of a summary score across all these different variables. And so this is using a Mahalanobis distance. This is the distance in multi-dimensional space across these 4 variables. And this is a method that is potentially scalable to an n-dimensional space. In this case, it's only 4 variables, but you could do it with 60 or 100 if you wanted to. It's basically looking at the sort of principal eigenvectors of each of these distributions and then seeing like how far an individual is removed from that in multidimensional space. So it's basically a very fancy way of sort of taking an average across those different variables without having to worry about one deviation in one variable kind of skewing your average. And then we use that to basically have a sort of little ranking to see across these sort of major clinical groups that we've looked at, which ones seem to be sort of the most atypical, at least in this sort of limited 4-dimensional space. And we see that Alzheimer's disease and MCI show by far the biggest deviation, followed by sort of schizophrenia, autism, and ADHD, but actually the anxiety and depression groups, they don't seem to deviate significantly if you look at it across all of these phenotypes together, although there is variation if you look at some of them individually. And then more as a sort of proof of concept, we also used both this sort of summary measure as well as the original sort of raw measures to see if they tend to show a stronger sort of heritability signal compared to when you would just use the raw measure. So using centiles, does it provide a little bit of sort of noise reduction in the signal effectively if we want to do something where we associate this with genetic variables? And for that, we use two data sets where we have a small twin sample, so ABCD and the Human Connectome project. And we do find that there's a significantly increased heritability of centiles compared to the raw values from which they're derived. So to me, that is effectively just telling me that by moving them to centiles, we're accounting for some of the potential technical variation that is still there in the raw data. And obviously, this isn't an entirely fair comparison, because usually when you would do this on the raw data, there'd also be some kind of harmonization, filtering, quality control, and processing that probably also takes care of some of that noise. But it's more a proof of concept to say that actually this might be a sort of viable way to do imaging genetics by using these standardized, somewhat harmonized phenotypes. That it might also be telling you a slightly different story than looking at the raw phenotypes. Now, there's a lot of elements in this paper that I've kind of glanced over and that in our case ended up to a large extent just in the supplements, but I think they're useful to highlight. So one of the things that we obviously had to deal with by stitching together over 100 studies is how do we actually account for this sort of variability across the studies and this technical variability. In our case, this was, I'm hesitant to say as simple as, but effectively we just included site by study or study. We did both, ended up the same result in our modeling framework, and basically allowed GAMLSS to figure out like to what extent it had to adjust the mean standard deviation at different parts in the lifespan for the different data sets so that they were actually like harmonized. And so what I'm showing in the left plot here in the sort of middle framework, this is just an example on the ABCD data set where we have 22 different sites. In the top panel, actually, you see what the raw values for gray matter and white matter look like. And you see there's considerable variability, even though all these individuals are roughly the same age range. They've all been acquired with a harmonized acquisition. They should be relatively well matched in terms of gender, etc. They're quite big. There's about 3 to 600, I think, per site, depending on which side you're looking at. But you can see there's quite a significant side effect, and there's a lot of variability across sites. In the middle panel, we're showing what it looks like after you've sort of used GAMLSS to derive centiles and then use those centiles to adjust the raw scores, and you get the side effects completely gone. So there's no significant difference anymore between the different sites, but there's still a lot of individual variation. So we're retaining the individual variation, but you can think of it as effectively sort of moving those box plots a little bit up and down to kind of level them out. What I'm showing in the bottom is combat, which is, I think, by now a very well-known technique to harmonize across sites. I don't think we're not in a competition with combat. I think it's an amazing technique as well, and it's suitable for a lot of scenarios. I think for our scenario, it wasn't that suitable because we're often dealing with a single variable, and combat tends to try and make use of the fact that you have a bunch of different variables that all have the same sort of tendency of side variation, and it tries to figure out, like, based on that sort of multidimensional framework, how it needs to adjust. That wasn't really applicable for us. The flip side of that is that in our case, we need quite a lot of data to do this robustly. So say if we had sites where only 5 individuals had been scanned, like our GAMLS approach would probably struggle to figure out like how much you would need to adjust because it doesn't have a good reference point. Whereas combat, if you have lots of variables but not that many subjects, still does a pretty good job at accounting for this. So I think both of these techniques are useful in different scenarios, and we're currently already working on figuring out how we could potentially actually combine some of these two things so that we have the most flexibility in terms of what data sets we can actually include in this modeling framework. The other element which I've completely ignored in the work here is always this element of having longitudinal data. So the idea that we want to make sure that what these centiles are picking up is actually consistent or is stable. So if you're measuring someone that is on a typical trajectory and you measure them, say at year 1 and they're in the 50th centile, ideally when you measure them a couple of years later, they're still in the 50th centile or somewhere around there, but if they suddenly jump to the 70th centile or the 50th centile or below the 50th centile, either there might be on an atypical trajectory or something is wrong with your measurement and you haven't properly accounted for the fact that there might be variation in technical variation in the measurement. So luckily enough, we had what we call a tiny subset. It's about 20,000 subjects where we had multiple measurement points. So this varied from just having 2 measurement points to, I think, the longest we had is someone, I think, had been scanned about 11 times over a period of 10 years or something like that. And in order to get a sort of sensible score that wasn't really dependent on the number of follow-up points we had, we basically computed the interquantile range of our centile scores in these longitudinal individuals. So this is more or less saying, like, to what extent does an individual sort of move up or down around their original starting point in this case. And what we see is that across all the different clinical groups and in the control group, this tends to be below 0.05. So there's less than 5% variation around the centile line in longitudinal data. So for us, this is a very nice confirmation that probably our models are quite robust, actually saying something meaningful about longitudinal data. It's also interesting to see that most of the clinical cohorts that we have tend to be even more stable. So there's 2 explanations for that. One is the statistical, the fact that these people are treated as a separate group means they might have less variation to begin with, simply by virtue of the modeling framework. I think the equally likely explanation is that once these individuals have a diagnosis, they're kind of stuck on that sort of trajectory, and there might be less variation from that unless they somehow magically recover or sort of jump back to normal, or back to typical, I should say. I think both of those are probably equally likely explanations, but regardless of what the explanation is, and regardless of the fact that there are some significant differences, I think the main message is actually that variability is very low, which bodes quite well for sort of potential future applications to longitudinal data. Though I should also say we're already also thinking about and working on ways that we could actually incorporate longitudinal into the modeling in the first place. Like, we didn't do that initially because it's less than 10% of our total data set, and any longitudinal effects would be completely washed out by the fact that most people don't have longitudinal data. But in an ideal scenario, we would be able to incorporate that. And then the last data slide I have is the one here on the last data figure I have is in the bottom here. We were also very lucky to have a tiny subcohort of about 300 individuals that transition during their longitudinal imaging. So they go from, in most cases, they go from not having a diagnosis to getting a diagnosis of Alzheimer's or MCI. And in a large proportion of cases, these are individuals that first time they measured, they already have a diagnosis of MCI, but they transition to Alzheimer's disease. So let's say there's an increase in severity of neurodegeneration across these 3 groups. And so what I'm showing in this plot is basically their sort of zero-centered centile scores. And you can see that for all of these groups that transition, you see that there's a relatively steeper decline in their centile score. So they tend to decrease faster than you'd expect purely based on the typical trajectory, which is, again, I think this is largely just a confirmation. But it might also bode well for this idea of maybe using some of these centile scores to detect early changes, maybe even before symptom onset. So that, I think, for me is also a very interesting sort of follow-up question, is to see, like, at what point do these individuals start showing symptoms, and maybe we can already see a decline in their centile scores that kind of precedes that. I think the other thing that's more of a research note to sort of pick up on, I think the dotted line here is the sort of typical trajectory from the full data set. So it's a typical trajectory from all the neurotypical individuals we have in the data set. And the thick black line are the neurotypical individuals in the data sets that also had these other individuals in them. But these are the ones that did not change diagnosis. So there were neurotypical the first measurement point, and there were still that at the last measurement point. And as you can see, that line also declines a little bit more than the total control cohort, which I think is interesting. And to me, the most likely explanation of that is that because these individuals are recruited as part of a neurodegenerative cohort or a neurodegenerative study, there might be a little bit of whether it's a bias or whether there might be family history in there. There might be all sorts of reasons why these people might be sort of a little bit closer to the clinical cohort than the sort of worldwide control group, if you can call it that. So I think that's interesting to pick up, and probably like the much fairer comparison would be to that group, because they're a little bit more homogeneous in terms of their recruitment, acquisition, and other elements of it. Now the bigger sort of last element of my talk basically relates to data sharing, and to some extent, maybe we can even call it open science. And that's the idea that obviously we collected all of this data, we processed all of this data, and in an ideal scenario, we'd just be able to say, here's a website link, feel free to download everything. You don't really need to redo all this processing because we racked up about 1 million computing hours just to process this data, and ideally no one would have to repeat that again. Unfortunately, we can't do that. Like most of the data that was shared with us is kind of shared under a specific usage agreement, different restrictions. In some cases, people might still want to use the data for their own project before someone else does it. That's obviously always a factor to some extent. But we still want the people to be able to use not just our data, but mainly to use our models and kind of use these normative curves as a benchmark. And so what we ended up doing is we ended up building essentially a model that is also a little bit more computationally efficient, because every time we run this gamma LSS model for all these trajectories, it also takes several thousands compute hours to actually do all the sensitivity and robustness analyses around it. And that's not very user friendly. If you tell someone, yeah, you can upload your data here, but you have to wait a couple of weeks to get your data back, that's obviously not ideal and probably restrict usage for a lot of people. So what we instead came up with is this approach that tries to sort of use a maximum likelihood estimation for a new study, so data that's not been included in our model, try and figure out how much it needs to adjust the sort of mean, standard deviation, and all these other parameters that we have in our model, such as the site, the sex, etc. How much does it need to sort of adjust that study for it to kind of fit in our normative curve? And once it's figured that out, it can compute the centile scores relative to that normative curve. So this is what we call our out of sample estimation. And we've made this available on our website, where people can basically upload a spreadsheet of their variables and then get back centiles for all their scores. The one big caveat I should probably put with that is that this works quite well if you have a data set that has about 100 or so controls, because we need a decent number of controls to be able to do that parameter offset estimation. That's obviously like currently a bit of a rate limiting factor. Again, this is something we're trying to solve to see how far we can bring that number down. Luckily, though, it would also work if there is legacy data. So, for example, in Cambridge, if the patient group that you're looking at has previously been scanned at the WIBIC or the Wolfson at the Wolfson Brain Imaging Center, there might actually be a data set that you could aggregate of 100 subjects that have been scanned over the past 10 years with a similar sequence that we could use to sort of compute these. So I think there are already possibilities in terms of if there's legacy data available, but we're also working on sort of bringing that number down. And then now, as I promised, I have a little video. I'm hoping this is going to work. So just shout if you can't see anything moving on the screen. But we've put all of this on the website. So first of all, you can just sort of explore the different data sets that we have, some of the basic demographics and basic characteristics that we have in our data set. As I said, there's lots of other sort of clinical labels that we have in this data set. You can select all of those, look at the demographics. To some extent, you can compare them. I think that comes in the next tab. So you see like what the age differences might be between all of these different cohorts. We have a little map of all the studies we've included. You can click on each individual study, and there's a link to the website, usually of the study, where you can request access to it. So even though we can't give you the data, we can at least tell you where to get the data. Then we have obviously these reference charts in an interactive manner, and you can basically click on the different phenotypes, and all these figures like change interactively. You can zoom in on specific parts of the lifespan if you wanted to see what they look like. For these regional volumes, we have a little interactive plot where you can kind of see like which regions sort of mature earlier, which mature later, with the different values with that. And then I'm thinking and obviously we can do the same sort of case control comparison that I showed you before, but we're doing it in a slightly, I would say, statistically less advanced way, because this is all run on a web server, and so there's a bit of a. I'm going to quickly pause it there for a second. So basically, this plot you see on the bottom right here is very similar to the plot we showed in the paper, with the only difference is that this is a very simple ANOVA or t-test to compare two different groups to each other, and it's not a more computationally intensive like permutation test as we probably do for an actual research study. In the top plot here, you can actually see in the shaded area, you see the sort of normative reference, and then around that, you see the raw data plotted from all these different clinical labels. As you might recognize, some people seem to fall way outside the sort of 95 centile lines. That is simply because this is plotting the raw data that's not been adjusted for like the study offsets, for sex, for other things. So it's purely raw, and obviously that needs a bit of adjustment. So very likely there's a study here that just had to sort of been pushed down because that study has a bit of a tendency to overestimate something. There's probably a cluster here of a study that tends to underestimate things. What you see in the bottom left is the actual centile scores that are adjusted. So what you're seeing here is a summary of all those points that have been adjusted for all those different offsets. And so this is actually something you could probably use for further downstream analysis. And obviously all these figures are also interactive. You can play around with these, you can compare them, you can use them for if you want to do some pilot analysis for a new study, or you're comparing to clinical diagnoses that haven't been compared before. You can probably use this as a sort of like proof of concept or a sort of first pass analysis. Now, as I said, we also have the option of you uploading your own data, and I think by now we have a slightly better frequently asked questions and template documents that tell you exactly how it needs to be formatted for you for it to work. And then there's this input-output tab that basically generates the same plots that you've seen in the diagnostics, but not based on our data set, but just purely only showing your data set. So this is a dummy data set that I think is also downloadable as an example, so you can kind of see what's in there. So this is a data set that I think is a couple of different, I mean, it's all made up data, but it's a data set that contains a couple of different diagnoses. So diagnosis 1 and diagnosis 2. I don't think these are this is actually a real diagnosis of any kind. I think these are all just random variables. And then it does the same thing. So it plots the raw scores like on top of the reference points. And again, these are not adjusted for a site or not adjusted for anything. It's just to get a rough view of where your data kind of sits. And then in the lower left panel, it shows you the centile scores. And in the right, you actually can look at some of sort of potential differences between the different diagnostic labels in this case. So on the left, it's just female and male, and on the right, it's by diagnostic label. You can visualize this, you can download the figures, you can, I think, download them in high resolution, you can zoom in, but you can also just download your own centiles back. So once you've uploaded uploaded your own spreadsheet, and then you subsequently click on down, this is just to show, you can also change this and the figures are updated if you change the phenotype. I'm not sure if this is shown, but at the very end, you can basically download your own centile. So you get your own spreadsheet back with a bunch of columns added to the end, the last column just saying centile score for all your subjects. And then you're free to use that however you like. And then that's it, I think. So there's just a final slide of sort of lessons learned, I think, throughout this project that I always tend to share, because I think these are hopefully all positive messages. So one of them is that there's already a lot of public data available. And I think even though we have now about 150,000, we're really only scratching the surface of what is available in imaging. So to me, one of the most exciting follow-ups is actually being able to include clinically acquired brain images in this, because there are literally millions of those just sitting on PACS systems in different hospitals. And that would also probably make it bring it much closer to clinical relevance, because obviously our reference is now kind of based on research study data, which isn't necessarily reflective of the individuals you might see in clinical practice. And obviously, as I've said, is not even geographically or ethnically probably very representative of the population. And so possibly getting access to clinically acquired imaging could remedy some of that. The other thing we've definitely learned throughout this whole project is that people are generally happy to share data, as long as you don't come with a whole watch list of variables and fine-grained things, and the sort of threshold for them sharing is quite low. So we started with very simple phenotypes. I don't know how this is going to go if we come back and say, well, we want this in a really high resolution atlas of some kind. I think people see the value of this now, which kind of helps people become more open-minded about data sharing. And since the papers come out, we've actually had various people reach out and say, oh, actually, if you're doing an update, can you include our data set in the next run? And people sort of basically offering up data, which has been an amazing experience. I also think we can actually, even though I keep saying these are very simple features, I think there is clearly a lot of signal even in the simple features in terms of whether it's enhanced genetic signal or just even looking at sort of basic case control differences, we can see quite a lot. I've already noted a couple of things that I think are super interesting follow-up projects, and if I had that like extra 24 hours in the day, I'd probably take on all of those, but I don't. So if there are people that are interested in doing some of these, I would also just encourage you to reach out, and we're always happy to try and facilitate if people want to follow up on specific things. then there's some buts and ends. I think these are all still positive. I think all of this requires community effort. Like none of this would be possible without actually having that massive group of collaborators and people willing to share data and expertise and knowledge. The big sort of caveat is, as I said before, is that this data is still very restricted to sort of Western, majority-wide, probably city-based, higher economic demographic, because these are the people that tend to be part of research studies, and that's something that I think requires a bit of a culture change. I think to some extent that's happening, like initiatives such as ABCD at least try and have a more population representative sampling and explicitly try and recruit for that. And I think that's definitely something you want to sort of take on board as you're designing studies. And then the last thing is like, yeah, you need big IT infrastructure to do these projects. I think we're now close to probably 2 million CPU hours of just processing, and we're sitting on close to 1 petabyte of imaging data. That's obviously not something that anyone can just have in their basement. So I think I'm quite lucky that Cambridge and also Penn have these facilities to sort of do this type of research. So the final slide is just to say there's been like a tiny core team that I mentioned, but obviously there's over 200 people, and this is also a group that keeps expanding. I'm not even sure if this is still up to date, because people keep willing to share data, help out, or just approach us for follow-up projects. And with that, I'm going to end my presentation. I've even managed to keep it within the hour.
Brilliant. Thank you, Richard. That was a fantastically clear presentation.